Hi, everyone. My name is Sarah Timmers, and this is my presentation on the article Genome-wide Meta-analysis for Severe Diabetic Retinopathy. The authors are Michael A. Grassi et al., and let's get started. So the question that this article addresses is, what genetic loci, also known as SNPs, are associated with diabetic retinopathy? So why do we care? Because diabetic retinopathy is the world's biggest cause of permanent vision loss in adults. Because most diabetics will develop this at some point in their lives. And this has a huge impact on the economy because of healthcare and labor costs. So by investigating the genetic cause, maybe something could be done to prevent it and treat it, such as drugs or CRISPR gene editing or things like that. So for some background, what is diabetic retinopathy? Well, it's a condition in people with diabetes that can cause blindness. It is the world's biggest cause of permanent vision loss in adults, as I said, and almost all diabetics will develop this at some point in their lives. The general cause is damage of the blood vessels in the tissues of the back of the eye or the retina from too much blood sugar. There are two forms of diabetic retinopathy. There is diabetic macular edema, DME, or proliferative diabetic retinopathy, PDR. So let's go over what these things are and what the difference is between them. So diabetic macular edema, DME, this is when small bulges called microaneurysms protrude from the blood vessel walls. These leak and ooze fluid and blood into the retina. This fluid causes swelling, which is the edema, in the central part of the retina, which is the macula. And you can see that in these diagrams here. Proliferative diabetic retinopathy, PDR, on the other hand, is the more advanced form of regular diabetic retinopathy. It is when circulation problems prevent oxygen from getting into the retina. This causes new fragile blood vessels to grow into the retina and into the vitreous, which is the gel-like fluid in the back of the eye. So you can kind of see here in these diagrams, the growth in the second one compared to the first one of the blood vessels. So what does this have to do with genetics? Well, we can see that the severity of diabetic retinopathy is associated with how much glucose is in a person's blood, how long a person has had diabetes, but also genetics. And the reason we know this is because people who have had diabetic retinopathy tended to have family members, siblings, or identical twins who had it as well. So the methods we use to study this are GWAS, which stands for Genome-wide Association Study. Two groups were studied, the GO-KIND group and the EDIC group. The groups only had people with type 1 diabetes, so there wasn't type 2 diabetics in the study. The GO-KIND group was genotyped using the Affymatrix SNP array 5.0, which is a type of SNP chip. And the other group, the EDIC group, was genotyped using the Illumina human hap 550 SNP chip. So to identify who had diabetic retinopathy, the GO-KIND group was given a questionnaire asking about their history of laser treatment for diabetic retinopathy. The reasoning for this was that patients must have ophthalmologists confirm that they have diabetic retinopathy before they can undergo laser treatment. So whoever was treated must have had diabetic retinopathy. The EDIC group was addressed by photographing the fundus, which is the part of the eyeball opposite of the pupil. And the reasoning for this was to look for signs of diabetic retinopathy. And if there were signs, they must have had it. So how does SNP chip works? Well, we learned in class that you have a sample of DNA and you want to know the identity of a SNP at a particular location. So you fragment the DNA, you cut it into pieces, you add a fluorescent label to the fragments, and then the chip has two types of oligonucleotides, which are short strands of DNA covalently attached to it. And some are a complement of the SNP allele, and some are a complement of the other SNP allele. So you add your sample of DNA that we fragmented to the chip, and it will bind to whichever oligonucleotide is its complement. Then you wash away the unbound fragments, and then the sample will bind to both kinds of oligonucleotides, but more will bind to the one it is complementary to. So more fluorescence on one side where more sample is bound indicates which SNP allele is present. And as you can see in this diagram here in the corner, the brighter parts, like where it's more red or green, that shows the side where this oligonucleotide is, where the SNP is located. So we did calculations as well. So again, in class, we learned that the odds ratio of every SNP in the genome was calculated to see which ones have the biggest effects. For each SNP, the p-value was calculated to see which ones are the most significant. And these p-values were then plotted on a Manhattan plot. Also, for the calculations, we had a control group. Two sets of calculations were done. There were calculations for all people with retinopathy, and then there were calculations excluding people with nephropathy. Now, nephropathy is a deterioration of kidney function, which is most commonly associated with diabetes. This exclusion of people with nephropathy was to control for the possible influence of nephropathy on diabetic retinopathy. So the results of this was, so first, here are the results for all retinopathy subjects with and without nephropathy. This is a table of the most significant SNPs that were found from GWAS. So there was a lot of information on this table, so I kind of explain it here, and you can look closer at this, but the most important thing we're going to pay attention to. So the alleles is tells what two base pairs are for that allele. So which, two base pairs. The chromosome is which number chromosome in the genome the allele is on. The genomic location is the base the SNP is in the genome, what number base it's on. The gene is the name of the gene the SNP is on, and left gene 3' to 5' tells which gene is closest upstream of the gene the SNP is on, and right gene 3' to 5' tells what gene is closest downstream to the gene the SNP is on. The GO-kind reference allele frequency is the frequency of that allele in the GO-kind group. The GO-kind OR is the odds ratio of the SNP for the GO-kind group. The EDIC reference allele frequency is the frequency of that allele for the EDIC group. The EDIC OR is the odds ratio of the SNP for the EDIC group. The Z-score is the SNP's relationship to the mean of the group SNPs in terms of standard deviation. The GO-kind p-val is the p-value of the SNP for the GO-kind group. The EDIC p-val is the p-value of the SNP for the EDIC group, and the meta p-val finally is the p-value of the SNP relative to both groups. So here's a table again. And the Manhattan plot, here it is for, it corresponds to the table that I just showed you. And it has the results of the GWAS meta-analysis of all retinopathy subjects. So each dot represents a SNP. The x-axis is chromosomal location, and the y-axis is p-value. So what was discovered was that 3 loci have a p-value of less than 10 to the negative 6, which means they're significant. And the most significant SNP associated with diabetic retinopathy is the SNP called RS476141, with a p-value of 1.2 times 10 to the negative 7, found at 242 megabase pairs on chromosome 1. So this is the results of the control group with the nephropathy subjects removed. And this is a table of the most significant SNPs that were found, and the headers are the same as I explained before. There wasn't a Manhattan plot for this group, but what was found was that there was no signals that overlap for the top SNPs that had a p-value of less than 10 times 10 to the negative 5, so there were no huge conflicts in the data. Of those top SNPs, none of them became smaller or achieved genome-wide significance, whether nephropathy subjects were included or excluded. And then in this control, The most significant SNP associated with diabetic retinopathy is a SNP called RS227455 with a p-value of 1.6 times 10 to the negative 7, found at 165 megabases on chromosome 6. So in conclusion, we discovered that in the group with all subjects, the most significant association for diabetic retinopathy found using GWAS is the SNP RS476141 for a p-value of 1.2 times 10 to the negative 7 and found at 242 megabases on chromosome 1. In the control group, the most significant association for diabetic retinopathy found using GWAS is the SNP RS227455 for a p-value of 1.6 times 10 to the negative 7 and found on 165 megabases on chromosome 6. So what questions does this answer? This tells us which SNPs could be responsible for diabetic retinopathy and where those SNPs are located in the genome. And what questions remain? One of the biggest questions is what SNPs are associated with diabetic retinopathy in type 2 diabetics? Is it the same SNPs as in type 1 diabetics, or is it different? That will need to be found out in another study. And that was my presentation. Thank you for watching. Bye.